This is part 2 of the fungi lab. Okay, there we go. I want to review what we did on the life cycle of the Basidiomycota. Alright, so this is a mushroom, and the mushroom is the fruiting body of a Basidiomycota fungus. The mushroom itself, I didn't write it, but is called the basidiocarp. That is the mushroom part of that organism is called the basidiocarp. And then underneath the mushroom, there's usually a very extensive network of little thread-like structures called hyphae, and all together the hyphae are called the mycelium. So we started out with the basidiocarp gills have structures in them called basidia. So we started out with how the basidia form within the gills. So the first thing that happens is a zygote forms from the fusion of two haploid nuclei and becomes a diploid zygote. And that diploid zygote divides by meiosis into a basidium cell with 4 nuclei. And then that is going to grow into the basidium, into the structure of the basidium. And the basidium is going to produce the spores. Just by cell division, it's going to produce the basidiospores. The basidiospores are haploid as well. And since there were 4 nuclei, there are 4 haploid basidiospores. Then we have dispersal and germination. And then we had the positive and negative mating types. We represented the positive with the red nuclei in the cells. The cells just grow into thread-like structures by dividing right next to each other. And you have 2 cells that end up fusing and forming, this is called plasmogamy, forming individual cells with 2 nuclei. Okay, so these are individual cells with 2 nuclei, 2 haploid nuclei that have not fused. So I'm just going to continue on with this and make one more cell with the negative nucleus and a positive nucleus, and then we will talk about the next step of the life cycle, which we're almost finished, actually. The next step is that these structures divide by mitosis into the hyphae underground and the basidiocarp above ground. So they divide by mitosis, which means the cells that form this structure here, the mushroom, the hyphae, this whole entire organism, the whole fungus is the combination of the mycelium underground and the mushroom above ground. So all the cells that form that structure are dikaryotic. They're dikaryotic. And dikaryotic means they contain 2 nuclei. So the cells, individual cells, this is how I'll draw it, have a nucleus from the negative mating type, we'll color that blue, and a nucleus from the positive mating type. And then what happens is called karyogamy. Karyogamy. That's this process right here. That is where the 2 nuclei, one from the positive and one from the negative mating type, the 2 nuclei fuse. So all of the cells in the mushroom have 2 haploid nuclei in them, and they fuse to form a zygote, a diploid zygote, and the cycle continues. Okay, so this is a sexual reproduction life cycle, and the Basidiomycota, the mushrooms, only undergo sexual reproduction. They do not have an asexual reproduction phase in their life cycle. Okay. So now we're going to do the zygomycota which is easy to draw in the fact that you don't have a large structure like a mushroom that you need to draw. But what's hard about it is there are two life cycles. There's an asexual and a sexual. Okay, let's see. That should work. All right. So what we're going to do now is we're going to talk about the life cycles of the zygomycota. An example of a zygomycota is black bread mold, and there is also another example of zygomycota that you're going to learn about through the videos that I post that I want you to watch by Wednesday. So I like starting with the same thing, with the same area, so that you can understand the zygomycota are just hyphae. I mean, their structure are basically hyphae, and then they grow into a sporangium on a stalk, and the sporangium is going to produce the spores. So you have hyphae, and then you have the little black bread mold structures. Like if this is your bread, I guess we can draw it three-dimensional. I don't know why I did that. There's no real reason. That's our bread, and our mold grows out of it like this. If you look at it up close, it's little black tips on stalks that kind of grow up above, kind of grow up out of the bread. So they're very small. You can see them easier with a microscope than you can with your naked eye. So anyway, let's start with those. We'll start with those hyphae, and it's probably easier in this case to start with asexual reproduction. Okay, let's do that. Let's start with asexual reproduction. In asexual reproduction, we're going to have mycelia formed by these haploid cells. There's no positive and negative mating type. What they do is they just basically like one cell breaks off and forms into a spore. Okay, so these are spores, and they're haploid, of course. And they're formed by asexual reproduction. They grow by mitosis into the mycelia, into the thread-like structures. So this is mitosis. That means they make copies of themselves, and those cells form into thread-like structures. So the spores germinate and grow into the mycelia. All right. Now, if those spores germinate and they form the positive and negative mating types, then we have a similar but different situation. Well, I'm going to draw 5 cells again. So this is going to be our positive mating type. So again, we will use red. Haploid nuclei. But the cell that comes out, that grows out, is going to have 3 nuclei in it. And then we will draw the negative mating type. And this cell that comes out is going to have 3 also. 3 haploid nuclei. Okay, so when these fuse, the structure that forms from the fusion of this is called, this area right here fuses, okay? And this fusion is going to result in a zygosporangium called a zygosporangium, which contains multiple nuclei. It's going to contain nuclei from the positive type and nuclei from the negative type. It's not necessarily a certain number, but there's going to be many haploid nuclei because of the fact that the cells that fuse to form that zygosporangium are multinucleate. Okay, so once that what that zygosporangium does is, and by the way, again, whenever the nuclei fuse, this is another example of karyogamy. Whenever nuclei from different cells combine into one cell, karyogamy. Nuclei fuse to form a zygote with multiple diploid nuclei. So now what's going to happen is this structure, the nuclei are going to fuse again. So you can guess what's going to happen here is blue and red make purple. So this structure is going to have two purple nuclei because the red and blue fused, and that comes out to be two diploid nuclei. Okay, so the zygote, again, this is the zygote, is 2N, and it is going to grow the positive and negative mating types This is going to be our positive, and this is going to be our negative. So they're going to grow from the zygote, and then out from the zygote is going to grow the sporangium, and the sporangium will make it purple because it's coming from the it's coming from the dikaryotic cell, the cell that has nuclei that have fused from different organisms. So anyway, what's gonna happen now, or what I meant to draw is a stalk. A stalk grows up, and out of that stalk grows a sporangium. That's what this is called. This is called the sporangium. And it is haploid, and it is going to grow into spores. It's going to divide and become spores. So that is your life cycle for the zygomycota. They can undergo asexual or sexual reproduction. The advantage of the sexual reproduction is that, of course, there's diversity, so genetic diversity. So now you're going to draw the last three life cycles, and you can just Google these. Just a reminder, our groups are the basidiomycota, the zygomycota. We've already done those. On your own, you're going to draw the ascomycota, glomeromycota, chytridiomycota. You'll take a photograph of your notes and drawings, put them into a document, and upload them to the assignment.